you can see, those are the following information or characteristics that you should know about lipid. Classifications of fats are categorized by three, which is neutral, saturated, and unsaturated. Neutral is simple, but how do you know which one is saturated and unsaturated? By looking at their chemical bonds. So let me show you a bit how. This is saturated chemical bonds and this one is unsaturated chemical bonds. Okay, so to make it interesting and more understand about the chemical bonds, which one is saturated and unsaturated, I'm using an app on my tablet to draw the chemical bonds. Okay, so this is saturated chemical bonds. I'm using the blue one as hydrogen and the red color one as carbon, and these two is oxygen. Okay, saturated fat has one single covalent bond. This is this one. For unsaturated fat, it has two that we call, this is cis configuration and this is trans configuration. Okay, so this is a bit different. As you can see, we call it band molecule. Another one, we call it trans configuration because the molecule is straight, so between this one. Okay, so you can create a lot by using any apps. I'm drawing it on my tablet, or you can drawing it on your own on a blank paper. You can color it to make it more interesting to learn.
The phospholipid structure layer. Firstly, the picture shown hydrophilic head, that is polar. In the head is phosphate head. Then the color red or slightly pink hydrophobic tails, non-polar. That is 2 fatty acid tail. Then in left picture, this is more details about amphipathic lipids in layer of phospholipid. Polar group in phosphate red clay or slightly pink. Polar group have a phosphate. This is a triglycerol. 1 phosphate group, 2 fatty acid chains. The clay color white is 2 fatty acid. Polar, and the structure is polar head phosphate in it and non-polar 2 fatty acid tail. Part wax water repel lipids. The function is to prevent water loss, and then wax together with fat and fatty acid make feeder waterproof. Lastly, the cholesterol and understerol. Cholesterol, the type in animal tissue. The sterol is lipid with a rigid backbone of 4 carborings and no fatty acid tails. Example sterol, estrogen and testosterone.
Okay, in this part, we can use online simulator experiment when we are at home. This simulator, we can test for carbohydrates, proteins, and fat. Our topic, we select facts, and we can see there is 2 types of tests, which are Sudan test and paper spot test. For Sudan test, we click and drag the dropper from the concentrated Sudan bottle and click to drop it into the test tube contains coconut oil. Then click the test tube and let it shake. We can see the results and click on the inference icon to see the inference. Pink droplets appear in the solution indicates the presence of fat. And then for the second test, which is paper spot test, drag the hand towards the crush glass to crush the peanut. Click on the inference icon and translucent spot appears on the paper, indicates the presence of fats. You can repeat the experiment by clicking the reset button.
Washing your hands with soap, possibly the simplest way to flush pathogens down the drain. Molecules can interact with pathogens such as coronavirus, leading to their destruction. So, it is very similar the way we remove our stain from our clothes. So, in order to remove our stain, we put liquid detergent in the washing machine, and what happens in the oil stain are removed by the surfactants. Each molecule has two parts. The head is hydrophilic, which means it likes to interact with water, while the lipid tail is hydrophobic, which means it wants to get away from water to find similar water-avoiding particles. This is the basis of how soap works. When the molecules encounter lipid particles on our skin, the cells aggregate around them to form spherical structures called micelles, creating water-free environment. Similar things happen when they encounter envelope virus, such as coronavirus. Envelope virus carry their genome and supporting proteins inside a lipid bilayer membrane with the protein necessary for infection embedded in the membrane. In cells and virus, these lipids are packed tightly into two sheets with the lipid tails facing inward.
The lipid end of the soap molecules are attracted to the lipids in the membrane. They take the advantage of the presence of membrane proteins, which can perturb the neatly organized bilayer to insert themselves into the viral membrane. If there are only small amounts of soap, the soap molecules only loosen the membrane, but with more soap, they begin to create micelles around membrane lipids. They are also attracted to hydrophobic amino acids in the membrane proteins, extracting them from the membrane. The structural integrity of a virus is essential for infection, so washing your hands with soap protects you better from infection than washing with only water. And the longer you wash your hand, the higher your chance of destroying the virus.